canal treatment prevents extractions and maintains teeth for function. However, non-vital teeth risk abnormal biting forces, excessive wear and fractures. Regenerative endodontics is thus a great alternative to conventional endodontics. This review is jointly done by University of Malaya, Masa University, UKM, Quen Cryocop, Malaysia.
Most of the pulp regeneration studies focus on dental pulp stem cells, which are from permanent and deciduous exfoliated deciduous teeth. They are actually originate from neuroectodermal origin and are far more superior than other dental sources in terms of blood vessel forming and nerve fibers forming. Dental pulp stem cells together with gel-formed scaffolds in a pulpless animal canal have shown pulp regenerations of pulp-like structures, and there's no sign and symptoms or periapical lesions after that. However, later studies have discovered that actually the regenerated pulp tissue is other form of tissue, such as periodontal tissue.
One clinical study and another case series prove that stem cells are able to regenerate pulp in necrotic immature tooth with maturation of root development and bone healing without complications and shortened treatment durations as compared to the conventional methods. Two clinical studies found that the DPSC and SHED showed success in pulp regeneration complete with neurovascular bundle formation, apexogenesis in immature permanent teeth. Further randomized controlled trial with larger sample size and long-term observation periods are warranted.
Scaffolds provide the temporary substitute of extracellular matrix as an environment for stem cells and various signaling molecules. The scaffolds can be natural or organic, synthetic polymers, or inorganic. Besides providing mechanical support and facilitating the process of cell homing and their differentiation, the ECM are highly tissue-specific and regulate cell morphology and specific differentiation. The major limitations of donor scarcity could be minimized by using decellularized pulp from ECM from in vitro cell culture from xenogenic sources.
Decellularization of pulp involves both physical and chemical treatments. Physical treatment disrupts the cell membrane to release cell content for removal by subsequent rinsing. This is followed by chemical treatments that remove the remaining cell membrane debris, which otherwise could induce an immunogenic response followed by implant rejection. Conserving the architecture and composition of the pulp extracellular matrix should be prioritized throughout the decellularization process.
The low cell yield in an autologous setup is because the starting material is very low and requires additional subcultures, thus other tissues may be more ideal source, although different in the propensity towards a specific lineage. BMMSCs with dentine matrix scaffold were able to differentiate into odontoblast cells with processes penetrating into dentinal tubules. Stromal-derived factor 1 attracts endogenous BMMSCs to migrate to root canal. However, its isolation involves invasive procedures, unlike umbilical cord MSCs available in large quantities in worldwide stem cell banks.
Okay, one of the major caveats in pulp regeneration is the concern of rejection of cells. Although MSCs from different origin have shown the potential of differentiating into odontoblast cells or any other functional cells in the pulp environment, the actual scenario in vivo condition have not been revealed yet. In this regards, the usage of iPSC cells could help to produce patient-specific stem cells, and apart from overcoming this limitation.
Total pulp replacement involves host tooth pre-treatment, cell processing, and cell transplantation. Pre-treatment disinfection of canal using metronidazole and ciprofloxacin added with clindamycin, cifaclor, or amoxicillin may be supplemented by negative pressure or passive ultrasonic irrigation and laser irradiation. Dental pulp and exfoliated deciduous teeth stem cells appear safe. However, large-scale controlled clinical trials are required. In conclusion, success rate in matured teeth is unclear and pulpotomy does not regenerate coronal tissues. Instead, in irreversible pulpitis, use of intradental root canal pulp tissues may better regenerate the whole pulpal tissues in its original form and function. Thank you.